Thank you for coming to my virtual poster today. My name is Jason Pitarresi. I'm a research associate at the University of Pennsylvania, and today I'm going to tell you a story about how we came to uncover a newly amplified gene in pancreatic cancer that might be a driver of disease progression and confer a new therapeutic vulnerability. So in recent years, it has become apparent that KRAS is amplified in a subset of pancreatic cancer patients, and that these patients might be those who have the worst prognosis. So we asked the simple question, what about the adjacent genes that are part of the KRAS amplicon that might be drivers? One gene that particularly excited us was the parathyroid hormone-related protein, which is encoded for by the PTHlH gene. And this excited us because it has been shown by others in the field to be a potential marker of the squamous subtype of pancreatic cancer. So we first looked at the TCGA database, and we saw that a significant portion of patients had KRAS as well as PTHlH co-amplification. This was about a third of the patients that we confirmed in a second UT Southwest dataset. And that if we looked at DNA, RNA, or protein levels of PTHrP, in all three cases, those with higher PTHrP had decreased survival, suggesting some sort of oncogenic function. Now, we next looked in the metastatic setting through the COMPASS trial, and we again saw that patients who had increased PTHlH copy number gains were primarily enriched in the metastatic patients as well as the basal-like subtype of patients. Now, if we looked at the Bailey, Collison, and Moffitt subtypes that have been previously identified, we saw that the squamous, quasi-mesenchymal, and basal all had increased PTHlH expression, and we think this is likely due to the squamous specific transcription factor TP63 being a driver of PTHlH expression. Next, we set out to delete PTHlH in the KPC-Y mouse model, where we have a YFP lineage label as well as PTHlH deletion. I will note that we only got heterozygous deletion here because PTHlH is just too close to KRAS. We could not get homozygous deletion. Now, the first thing that was obvious from these animals was a dramatic increase in survival. So we had a nearly 80% increase in overall survival relative to KPC-Y animals, despite the fact that we only had a heterozygous deletion. Now, even though these animals were roughly 2 months older at endpoint, they had greatly reduced metastatic competency. So if we look at primary as well as metastatic tumor burden, it was decreased upon heterozygous deletion of PTHlH. So we next asked if we could therapeutically target this through monoclonal neutralizing antibodies directed against PTHrP. We did this in the orthotopic setting, where we implanted tumors, waited for them to grow 7 days, and then began therapy. And we saw that primary tumor burden as well as metastatic tumor burden was greatly decreased in the anti-PTHrP treated animals, and that even if we did a forced metastasis assay, such as a tail vein metastasis, and then treated with anti-PTHrP, we again decreased metastatic outgrowth. I would like to note that I've also performed shRNA knockdown as well as CRISPR-mediated deletion of PTHrP, and we get complete phenocopies in vivo, but I don't have time to show the data today. Now, finally, we wanted to get at the potential downstream mechanisms that are driving these phenotypes. So we first looked at the tissue of the heterozygous deletions of the KPC-Y animals, and we saw that a significant portion at the population level simply did not progress to PDAC. However, when we did compare the PDAC animals from the KPC-Y to the KPC-Y PTHlH heterozygous deletion, it was quite obvious, strikingly obvious in fact, that PTHlH deletion shifted the phenotype from primarily poorly differentiated in the KPC-Y to primarily well differentiated in the heterozygous deletion. This is perhaps most obvious when staining for a YFP lineage label as well as E-cadherin, whose loss would indicate EMT and beginning of the dissemination program. You can see when we quantify this that there is just greatly decreased number of disseminated tumor cells. Now, finally, we performed RNA-seq in cells lacking PTHlH, and one of the top pathways, the second most upregulated pathway, was the EMT pathway, meaning that all of the EMT transcription factors and target genes were enriched in the non-targeting controls relative to the PTHlH knockdowns. Now, finally, if we took these cells and tried to force an EMT through TGF-beta treatment, we noticed that we were unable to shift the PTHlH deleted cells into a E-cadherin low state, suggesting that they could not undergo EMT. So I think we've shown nicely that PTHlH is a new oncogene and potentially pro-metastatic cytokine in pancreatic cancer, and I'm happy to take any questions you might have today.